Oh, my apologies, my battery died, so we'll start with that again. We were saying in DNA replication, what happens is that the free nucleotides pair up with both strands. And what happens in this is we only have one strand that is the template strand. And if you think about it, that makes sense. Let's say that the bases for my eye colour protein were AAA. Let's say that was as much as the gene was. It's just AAA. Obviously, it's a lot longer than that. But let's say AAA made brown. Well, on one strand of my DNA, it's going to say AAA. On the other strand, it's going to say TTT, because T always goes with A, always bonds with A. So it wouldn't be any use copying the TTT strand. That's going to be gobbledygook. It's going to be meaningless. What we want is the AAA strand. Only one of the strands will ever be useful, and that is the template strand. So, the nucleotide bases on the template strand pair with free-floating complementary RNA nucleotide bases. So if on the template strand it said AAA, the DNA said AAA, well, they would pair up with three U's, U, because they're uracils, and they will bond onto the DNA. RNA polymerase, which has a very similar function to DNA polymerase, then catalyzes the formation of phosphodiester bonds between the new nucleotides. So in my example, I've got my AAA on my DNA side, I've got my UUU, which have now been attached. I'm going to join all those U's together. And then the RNA polymerase stops when it hits a termination sequence. So basically, it's going to drop off. And what you should be left with is a single strand of mRNA, which is complementary to the template strand. So if the template strand has an A, the RNA should have a U. If the template strand has a T, the RNA should have an A, and so on. And it's very similar to DNA replication. Difference is only one strand is copied rather than two. We're using RNA nucleotides rather than DNA nucleotides. We're using RNA polymerase rather than DNA polymerase. And we're only copying a very small section of the DNA, one gene, not the whole chromosome like we would in DNA replication. So, I'd like you to have a look at those three questions. To be fair, let me take this back so you can take the notes. Pause me, take the notes, and then we'll go on to the questions. Right, there's questions, three questions there. I'd like you to have a look at them. I'd like you to make sure in your own mind that you can answer them. So take a minute just to read the questions and make sure you have an answer. There's not really a need to write them down because you just wrote down the notes. Try to do it without looking back. Right, hopefully you paused me. The name for the process where mRNA is produced is transcription. The name given to the strand of DNA which is used for the RNA nucleotides to attach to is the template strand. The enzyme that builds the phosphodiester bonds is RNA polymerase. And it's just that simple. Now, there's one more stage at the end of transcription, and it's usually included as part of transcription. And that is that actually, that's not a final piece of mRNA. That is a pre-mRNA. In prokaryotic cells, actually, that would be the end. That would be the end. We'd have the mRNA, it would go off and do its job. But in eukaryotes, you have to modify the mRNA. So, we have our pre-mRNA in eukaryotes. I want you to have a look at that. This is how we're going to modify our mRNA. I'd like you, without knowing anything else, to tell me, firstly, what do the 5' and 3' labels mean? We're coming back to that again. And secondly, to describe the process of mRNA splicing in eukaryotes. Now, I don't expect you to know what an intron and exon are at this point. Can you just describe what is happening there in your own words? What can you see happening? Just have a minute, pause me, to write down those answers. So, the 5' and 3' labels refer to the ends of the DNA. Now, the 5' of the mRNA will actually have been on the 3' of the DNA. It's basically a way of orientating yourself. We know that most processes always run from 5' to 3', including transcription. So it just refers to which end of the mRNA we're looking at. The 5' will have a phosphate on it, and the 3' will not. To describe the process of mRNA splicing, what you should have written is something like this. We should have written that the introns are being removed and the exons are being attached together. Now this is called splicing, and the reason this happens is because there are sections of eukaryote DNA that are not necessary for making the protein. Sometimes that's just what we call junk DNA, DNA that doesn't do anything and so it needs to be removed from the mRNA. Sometimes it's bits that are required for enzymes to attach to, but are not actually useful for making the protein itself. For instance, there's a binding site for RNA polymerase, which RNA polymerase attaches to. That's not used for making proteins, so we have to cut it out. But regardless, the introns are the useless, unnecessary bits. The way I remember that is an intron is an interruption. That's the way I remember it. Now, the introns are removed by an enzyme. We're not going to talk about which enzyme it is. In fact, there are quite a few. And that's called splicing, allowing the useful bits, which are called exons, to be attached together. So we've removed the interruptions, the introns, and we've got the exons, which are excellent, if you like, and they are attached together. And this is all being done with various enzymes, which we're not going to go into right now. Again, please get down those notes if you haven't already. Right. Now, we're going to do some questions now on this. So, all we need to know so far is the basic stages of of transcription and the different types of RNA. So we're going to do some questions on each of those. That's your first question. It is, I believe, a 2 marker. I don't really need you to copy out the table in full. It's very similar to what we did earlier. Can you just please work out for yourself where would you put ticks and what numbers would you put in the number of polynucleotide strands? Again, hydrogen bonds present or not present? Well, that's an interesting one. DNA? Well, yes, definitely. Hydrogen bonds are present because we have these 2 strands with the A and the T and the C and the G attached together by hydrogen bonds. Number of polynucleotide strands in DNA? 2. mRNA, hydrogen bonds present? No, there are not. There are no hydrogen bonds in mRNA. Hydrogen bonds are used to attach nucleotides together. An mRNA is just a straight long line of nucleotides. We don't need to attach it together anyway. Number of polynucleotide strands is 1. 1 in mRNA. Hydrogen bonds present in tRNA? Well, yes. The way we get that cloverleaf structure is we get the bases to attach to each other. And when they attach to each other, that's a hydrogen bond. The number of polynucleotide strands in the molecule, though? 1. Only 1. The last thing we're going to do then, to be fair, no, it's not the last thing. This is a bit about splicing. This is a 3 marker, and I actually would like you to write this one out. So again, pause the video. 3 minutes, I don't think you'll need it, but pause it. Right. In the space below, give the sequence of bases on the pre-mRNA transcribed from this strand. So this is DNA. You can tell that because it's got T's in it. So the pre-mRNA we're going to make should look like U, G, U, C, A, U, G, A, U, G, C, U, A, G. Hopefully I got those right, or I'll get all kinds of comments. That's 2 marks there. Give the sequence of bases on the mRNA produced by splicing. We just remove the intron. We keep the exon. They're excellent. And it should say UGU, UGC, UAG. That's the answer. Fine. Incidentally, those 2 marks are for 1 getting the correct, on part 1, of 1 getting the correct bases, and 2 correctly replacing all the T's with U's. So let's say you put TGT on the first one, you would only get 1 mark because you forgot that then you need to have eurocells. Right. Okay. CTFR. Now, this is a bit more of an application question. Again, there are 2 marks. Can you please again pause me for 2 minutes, have a go. Simple bit of maths. My apologies, Year 12. As it turns out, we haven't actually done this bit yet. So I'm going to leave that there. That's my fault. So we'll leave that. We'll come back to it later on. Okay. That's the end of that. Now, what I need you to be able to do from this lesson is to be able to tell me what the difference is between mRNA and tRNA are, be able to tell me the stages of transcription, which are there, and to be able to tell me what splicing is and how it works. If you can do those 3 things, then you've understood the question. Now, I apologise for the extra exam question on the end there. We'll be doing that on a different lesson. Right, come back next week for translation. This is very important. It is important to understand it, particularly what's on your screen now. Please make sure you have memorised it. Hopefully see you soon.